Thank you very much all for coming to this talk this evening. We are delighted to have you here in this beautiful space at the Woodside. My name is Florian Carle. I'm the manager of the Yale Quantum Institute, and I curate a series of non-technical talks that try to mix science and the humanities to try to make science more accessible and less scary by bringing in some of the humanities folks. It turns out that sometimes it makes it even more exciting, but also more complex. But that's the beauty of that series. This series has been co-sponsored by the Frankie program that works with us for the longest time. We started this series in 2017 to try to see what we could do. We had many talks and many topics, bridging the gap between quantum physics and music, between physics and dance, between mathematics and crochet, space exploration and board games. So we had plenty of these things, and we're really happy to start another session tonight, mixing neuroscience and art. So I think it's the perfect place to be in tonight at the Woodside because their mandate is neuroscience and data science, and mixing these two to try to accelerate breakthroughs in understanding cognition. And so what a more beautiful and interdisciplinary way of bringing this and having two speakers tonight with us, the Italian artist and visual artist Serena Scapagnini and the neuroscientist Michael Higley for this talk. And so thank you. Let's give them a warm welcome. And so I've talked enough, so instead of doing a bio introduction, I'm going to let Serena start with an introduction.
Thank you. Well, first of all, I would like to extend my heartfelt gratitude to the Yale Quantum Institute and to the Woodside Institute for organizing this conference. And of course, to Florian Carle, manager of the Yale Quantum Institute, and to Professor Higley. Without his visionary openness to my research, many shapes, places, encounters, and artworks would be very different from what we are contemplating here today. And most likely, I wouldn't been here at all. So it's a joy to be here with you tonight and to share SYNAPSES, my most recent project, dedicated to the mind and to the synaptic connections and to the memory mechanisms. In this body of artworks, I've been working inspired by the brain cells, and I was looking for a collaborator to deepen through the imaging processes, especially the rhythm of the brain thoughts, specifically how the rhythm of thoughts branches out from the visual and the morphological aspects, and how this creates a sort of visual identity between thoughts, memory, and images. The artworks stratified between paintings and drawings upon an acid solution preparation, future branching neurons inspired by the living brain tissues. In this body of artworks, I've been working to dozens, then hundreds of drawings and paintings, especially on paper, but also video artworks, long scrolls, and architectonical structures, like installation made by the overlapping elements. So again, I want to thank Professor Higley for this collaboration, which runs since the 2012, and here we are.
Thank you. Professor Higley?
First, I'd also like to start out by giving great thanks to all of you for joining us tonight, for the Woodside Institute for hosting us, and really for Florian for making this evening possible, and the Yale Quantum Institute. It's really actually been quite an inspiring opportunity to see what Florian and his group have managed to do in the past, bringing artists and scientists together, and the hope is that we can continue doing something like that in the future. I owe really sort of profound gratitude to Serena for letting me participate in this. There are several scientists in the room, I know, and many of us spend our lives trying to nail down the details and honestly make things less ephemeral. In fact, in a way, that's sort of our job, is taking things that are ephemeral and in fact taking the mystery out of them and making them very solid and present. And I think, my interactions with Serena and her art, touching very lightly on some of the images that we provided, but all of the creativity is hers. But that experience has at least given me some opportunity to go in the opposite direction and maybe see how our science can lead to more ephemeral or more abstract ideas. And so that's been a great joy for me, and thank you.
And so to kickstart things, when we think about neuroscience and when we think about arts, that's not something we put together right away. And so can you tell us how you met? Like, how did the collaboration start? I know that your art has been exhibited in India, Milan, New York, all of these places, and Mike, you've been also dragging along all these places. Like, how did you two meet, and like what sparked the collaboration?
Well, thank you. As you said, my art has been nourished experimentally and conceptually by a substantial interdisciplinary approach since the beginning. Peculiarly at that point, I was working with memories and focusing my attention on the study of form. So I thought that through neurobiology, I can focus on these imaging processes to get into the vision of the brain cells from real. I wrote to Professor Higley with this idea to go further and to get experimentally closer to the subject which I was studying. Once the project was born, it was the 2012. Then from there, of course, we had many chapters, and the works has been shown in India for the India Art Fair in 2015. And then in Milan, in the US, I had a couple of shows in New York and in Miami for the Art Basel Fair. And then in a couple of days, it will be presented in Naples at the Pio Monte della Misericordia, which is a very prestigious and beautiful church in the heart of Italy. So, as I said, many shapes and many cells coming from these labs are sprouting around the world.
And so you're mentioning an email. What does it feel like to have an email from an artist coming in your inbox early on?
Yeah, I mean, it's quite remarkable. I mean, I think as many of you appreciate, I mean, we get a lot of emails, and some of them are very on point, and some of them are sometimes a bit, maybe out of left field. And I don't know. I couldn't tell you exactly what fraction of those maybe sort of left field emails really grab your attention, but for whatever reason, in 2012, I received an email from Serena looking for images and maybe a collaboration of sorts, and I wrote back, and I'm not sure I have any memory of a more pressing reason than that at the moment. I mean, it was I don't know, fortune or serendipity or whatever, but I wrote back, and we were sort of off and running from there. I mean, from initial interactions and trading images over email to getting to experience an opening of a show in New York, and later seeing pieces in Italy and other places. It's just been quite an amazing experience.
And so you touched a little bit on the creation process of exchanging email online of like the different brain. Like, can you tell us a little bit more about the creation process? Like, what drove all this creation of the artwork that we see in the back in the reel?
Thank you. Well, part of the process is substantially ineffable. But just to give you some technical details, I started working on the medium of my artworks. So recently I made the paper handmade in Fabriano, which is an apt place in Italy for this tradition. And this gives me the possibility to work with its peculiar chemistry. So I can add copper inside and colloidal gold. This chemistry within the paper is also putting all together some conceptual and physical elements. Then after that, the paper is treated with an acid solution to collaborate to this effect of rarefaction. Because in all this project, the dialogue between the cells and the images inside needs to survive, to have this void concept that is always creating a dynamics inside of the paintings. And with this empty white surface, I work a lot with acid to give us an interaction which is slightly aggressive with the paper, but gives me the opportunity to let the colors flow inside more deeply. And after this, I work with paintings, and of course, I am pretty faithful to the models, especially in terms of the mathematical proportions of the dendrites, the possibilities of the cells to dialogue respecting the space between the cells. And after that, I work with pure pigment dust and China ink just at the end. The China ink images float upon the paintings, and I try to be very respectful of the matter. So, in a sense, is a matter that gives rise to them. This is obviously part of the artistic process, you know, to take the images coming out from the matter. And in that part of the work specifically, I also work deeper about memories and how the flow of thoughts and the flow of memories interacts with the single image, in a way how a single shape interacts with the surrounding fluid and the surrounding structures. So this goes also in the direction of deepening the osmotic exchange between cells and more broadly between structures and space.
You know, maybe a huge defining part of not only our interactions, but of Serena's art, which she touched on, is the combining of things across different scales, maybe across different scopes. I mean, just one of the things that she mentioned is one of my most favorite aspects of her art, are the small China ink drawings that are at the end, because they're often representative of more, I think, complete ideas or memories or images. And they're quite small, but then superimposed on these individual neurons from our images that are sort of enormous, and so it's this inversion of scale almost of complete thoughts and memories, somewhat more ephemeral, to use the word again, on top of these larger, more concrete cellular structures. And for me as a microscopist, where much of what we do is trying to use devices to make things that are really small look big, and then at the same time have these large concepts, memories, images, and so forth, appear small. These inversions of space are really quite fascinating for me.
Once I stepped finally into the labs after more than 10 years that I work with your images, and to see the scale of the microscope and the scale of the slices of brain that you are interacting with. And of course, considering the inversion of the scale, that the neurons are becoming so huge, and memories as this flow of images, like an act of sharing between the visible and the invisible in a way, because what is the substance of memory, I guess is still not clear even to the neuroscientist, but is very poetical to wonder where are the memories stored, what is the substance of memories, what's happened when the memories get lost.
And you were talking about the sense of scale. Can you tell us a little bit about the scale of your artwork? Because I think for the audience, it might be a little bit hard, especially on the screen, to see what's the scale. And you often have like a synapse expanded, or like, can you tell us, give a sense of like how big your artworks are?
For example, these long scrolls is like 2.5 meters, and the main works on papers are on this scale, more or less. The very first work that I've been creating for a private collection in Italy at the Studio Synthesis was a 5 meter. This installation, which has been shown here in New Delhi, but then site-specifically adapted in Milan and Miami, it runs from 2 to 3 meters. I always keep the humans as the models of the scales of my architectonical installations. So as if the humans stepping into the overlapping elements can complete with their own field the artworks. So, for example, you saw also that toroidal shape. And yes, in this case, the scale is 4 meters per 4. So that if you put a man in the center of the two spirals, it completes it perfectly. So it's filled.
It's quite interesting because we're talking about the memory and how people like proceed, and so you basically enter your structure and you can have this artwork all around you.
Yes.
We lose a bit the sense of where memory starts, and because you have the very biophysical sense of the synapses, and then you have the human interacting with the artwork. And so how do you what can you offer to the public? Like, where do you draw the line of like we should stop the name of the talk is The Shape of Memory, and mm-hmm. you're shaping this artwork around it, and what does the fact that you have this architectural structure around the art provide that you cannot provide with just a painting or something? Like, why do you feel the need to bring that into a structure?
The architectonical structures and their systemic configurations allows me to evoke the shapes which underlying many shapes in nature. So as well as the field and the geometric symbols, which as a joke pass through the brain. So they are in a microscopic scale perceived by the brain, and then at the same time surrounding the viewer, as if thought could in a way create the world around us as a sort of a premonition of life, and as a sort of premonition of consciousness in a way. The fact that we always have the white surface on one side with the more plastic and visible heaviness of the images surrounding us. It's like giving an homage to the spectrum of the visible, so to the spectrum of our perception, and at the same time blinking one eye to what is over this spectrum, what is not catched exactly by our brain, but perhaps by our consciousness.
Yeah, thank you. And Mike, your job is to try to figure out how to image the brain. Can you run us through a bit of your research in your lab and like how you produce these images? Because we went through the artistic process.
Sure, absolutely. Now, some of my lab members are here, so they'll have to forgive me if I botch the description. But yeah, so I mean, for most of my career, my lab has been focused on trying to understand how the various bits and pieces of the brain interact with each other to create thoughts, to create behavior. And we work at quite a number of different scales. I mean, from individual brain cells and even, at a subcellular scale, looking at individual synaptic connections, all the way up to behavior. And so the lab is quite focused on trying to span a range of scales. One of the common features that ties a lot of that together that's perhaps an interesting coincidence with Serena's work as a visual artist is that we use light primarily to study brain activity. And so we're often shining lights of various forms onto animals and onto brain cells, and then collecting the light that comes off and using various means to interpret that as an insight into both the structure and the activity and the function of brain cells. And so, as we've been talking about, this is essentially using microscopes whose purpose is to make things that are very small look very big, so we can understand them. And perhaps as a nod to what we were just discussing as well about the size of the pieces, having seen many of them in installations, it's truly amazing to almost be able to step inside Serena's pieces as if one was stepping inside the brain, but less concrete than that, almost being able to step into perhaps what are ideas of what the brain is doing, and you can see some of these as they go by on the screen, getting a sense of what it might be like to be inside a memory or a thought, and so it's quite inspiring, again, to see things that are so concrete. I can look at some of the neurons that are in the paintings. Oh yeah, I remember where that was from, and it's quite transformed in this different context.
What was the benefits of that relationship? Because there's a lot of conversation about mixing art and science, breaking down silos, but what did it concretely brought to both of you?
Well, I think that the images from these labs, they talk themselves about this incredible harmony and beauty, which is in their also asymmetry and their growth and their, as I said, mathematical proportion. But technically, especially as Professor Higley was suggesting, the use of light is giving to my work the special tool to step into the movement. So creating artworks made by layer and layer of frames in movement. And on the video artworks, the light is really pumping out, coming out from the painting and crossing physically through the artworks without frame, actually, because the paper has copper cables inside, but also use the backlight to enlight some portion of the artworks. And so these lights, which comes out from the two-photon microscope, and from that to the images, which I can, of course, chromatically re-elaborate, but that I follow faithfully in its path. And then the light again is working on the artworks, both in the video, because the videos are running upon the artworks and lighting certain portions and creating this effect of enlightenment of certain drawings. And finally from the back of the artworks. This let me stay into that peculiar, what Leonardo da Vinci says is that there's something sharing between the veins of my hand with the branches of a tree, with the, and now we can say with the brain cells proportions. We find a deep identity between art, science and nature.
I think for me, it's been quite maybe a personal sense of not only exploration, but a little bit going back to, at least for me, why I started in neuroscience in the first place. I think I was very early on taken with the idea that 2 or 3 pounds of lumpy gray flesh in our skulls gives rise to these amazing experiences that we all have, some of which we share, some of which are unique to ourselves. And then you spend, like I said, you spend a career trying to nail that down into details. And I think, being able to not only, talk with Serena, but see some of that work reflected in the art that she created out of it is at least a reminder that there's something sort of miraculous beyond these details that we're figuring out in the lab. So,
thank you.
So I have a last question, and then we'll open up to the public. If you have any question, feel free to raise your arm. I'll come with the microphone. And so how did your work change after this collaboration? Like, it's an ongoing collaboration, but did you had another outlook on your work, something that you would have not necessarily done in your practice by this collaboration with the scientists, or how did it change for scientists to interact so much with the artworks?
Like I said, I don't know. I think especially the more senior along I get, the more opportunities to think, maybe broadly, maybe a little bit more abstractly about the work that we're doing, and just to appreciate the art in what we're doing. Actually, one of my greatest experiences of my graduate mentor was just the love of the beauty of the science itself. That just seeing a new result show up on the computer screen was forgive me, but almost watching a Da Vinci suddenly appear. They were just so amazing. And honestly, seeing Serena's art gives me a little bit of that sense again. So.
The inspiration that I get from the brain cells is, of course, the starting point. But then the most important aspect that I can see today is that the work is never stopping to go ahead and to deepen the subject. So I might say that the scientific process is giving me the possibility to work with an outer regard through the inner regard, which is obviously there once you practice with art, to enrich the work with this perception of the inner vision of the world within the brain was really fantastic. And so I'm deeply thankful for all this process that is growing all together with the friendships, I can say.
Hi, thank you so much. My name's Katie, and I'm from the Divinity School, so this is not my world at all. So I'm very grateful to be learning about this. I have a question about design choices, specifically around color. I see so much color, and it's all different in all of these images. So how did you choose the color? And also the images. Why did you land on some of the images that you did? What stories are they telling that you're hoping to convey, or is that even part of your process? Thank you.
Thank you. Well, first of all, about the colors. As I said, I use pure pigment dust, and together with the brain cells that I receive as models, which are original, I have substantially worked with 3 different layers of colors. The black and white that you see, which is more connected with the drawings and with the memories and with this also, we can say, stream of consciousness. And for these works, I use a lot of magenta and white combined with some mineral pigment, like mica minerals. And they are the more plastic works with the huge installations, with the space surrounding the viewer. While for the blue ones, I choose those colors because they are probably the more intimate works, and I wanted to work, of course, with memories, but also with the personal stories that belongs to the process. So in the blue works, more than in the red-pink ones, I have put a lot of drawings, as Professor Higley was suggesting. In those images, I think where I have to be very straight and dry in a way with myself is to takes a part of a personal history, but without following just my memories, in a way stepping to that archetypical source of memories, which are collective also. So in the blue ones, I worked more about this. So it's not just my memories or maybe my grandmother's memories, but there are stories that in a way are stored somewhere in the collective archetypical visions. And so I try to work more on this level so that they can be storytelling, not just to myself. No, it's more like being a channel for the images to come out.
I just want to ask to either of you if there's any, what a particular vision or hope that you have is for the future of such art and science collaborations as this. And the second kind of part is, I think I've noticed in my experience that there's not really a lack of mutual interest between artists and scientists, but systemically it doesn't really seem to be very well realized. I don't, especially from the science to art side, I think that there's kind of difficulty in, I guess, getting scientists more engaged with art, and especially like these kinds of practices, which I think can, you know, like you said, be really interesting to science in a lot of both like productive and also just like fun ways. Yeah, so I don't know if you guys have any thoughts.
No, those are great questions. I mean, I think maybe just to paraphrase and make sure I have it right, because I think it was an important one. Is maybe a little bit. Is it difficult, or how do we get the scientists to be more active participants in the process, or engage more? Is that a fair? Yeah, I mean, I think there is a challenge because in a way we're trained to give up all of that, right? I mean, we're like the art or the practice of being a scientist is to try to, ignore what you feel or want or think might be true, and just try to take the data that are in front of you and interpret little and maybe think much. But I mean, in a way, then I think what the art does is gives us a chance to maybe in a kind of a safe way get beyond that, to say, oh, I can think broadly or grandly because I'm considering a piece of art. I'm not considering a piece of data that I have to publish. And so I think there's real value in that. And maybe for me, early on in your question, I was also thinking, probably I couldn't tell you where most of my scientific ideas have come from. I mean, standing in the shower is a really common way where scientists will tell you where they think of stuff. And so it's almost anything that forces your mind to wander and go to places that it might not otherwise have gone, whether it's looking at the art or thinking about it or even talking to the artist and trying to explain what I do while getting a sense of what she's doing. There are kernels of inspiration in there that are difficult to say why they are or how they came to be, but they're there. So I think that would be a hope is for all of the scientists in the room to find opportunities to be more creative or find those kernels of creativity more readily.
There's a lot of overlap between the scientists, and many of them are artists themselves, musicians often, and so there's this spark of creativity that is there. One thing that was interesting about like how to engage more of the science towards the arts, there was a lot of pushback when I tried to create the Artist in Residence program, because the scientists had this vision of like the science, the physics is beautiful itself, and so why should we have an artwork inspired by science or after science instead of just a plot, which is the actual physics about the research we've been doing, and I think that's a good debate, because we could have just a plot, just one figure, but I don't think the pure science inspire. There's something that the interesting part about the collaboration between art and science, I felt, at least in my experience, that is, it's not just a fact. It's something that goes beyond. You can express things that the physics might not like be able to do. For example, we were talking about memory earlier. You can have as many photos of synapses as many, but it's not going to give you that feeling of we can think about it the same way that we could have as many plots as we can. It's not going to inspire you, and so maybe that's what we want to do. But the mix is giving like a stepping stone to go further in what being a human is, I feel, and that's what bringing the art will help, and that's what we're trying to encourage, and so.
Well, thank you. I agree with you. I just wanted to add this, that I think you're right. I mean, the beauty is already there. The artist perhaps adding the vision to that beauty. And on the other side is fascinating to collaborate with science, because art as well as science is working with probability. So once I said I have to remove myself to get the artworks straight and clean to the point, is letting this probability of the artworks to become what the artworks wants to be, like in a sort of autopoiesis. And is not science also working with this autopoiesis of nature? I mean, taking the information from something which is already there.
I wonder if you know about the thought, of the shape of the thought. Do you know what this person was thinking of that image?
Sorry?
What is the thought behind that image? Do we know, is there any information about that?
Which one?
Any of them?
The information.
Yes, like, is there, like, how are these images taken? Do we know, like, was this person watching a nice movie or a brutal movie, or like what could possibly have been the thought behind this and that
The
image?
actual data, you mean? The
Yes,
science.
and then do you as an artist know about that, and do you work with that information?
But so very quickly, but then I'll let you know. Just to clarify, so these are all mice. So all of the images are taken from mouse brain cells and mice brains, which is not to say I'm sure that they do have thoughts. It maybe in a way one might actually say. I mean, I'm quite convinced that they have some sort of internal, goings on that I think is forever occluded to us. I mean, this is one of the challenges with working with organisms that can't speak, is they will never be able to tell us, in their own words, what it is they're thinking. And so we are forced to apply some of our own, vision or images onto just the data. So we don't know what the mice are thinking. We could imagine, but
Portraits of mice, the shapes of neurons and the shapes of the dendrites, and especially of the spines, which I use as models, as inspirations, are there, and the images are potentially surrounding those neurons as like apparition. You can tell me a lot of our thoughts, perhaps even of animals, but we are not sure, runs through images.
certainly for us, I mean, I think we are very visual organisms, but I think the more we learn, probably most organisms with a visual system have a pretty robust visual sense and image-based sense of the world.
So we frame a single shape of images in this river of thoughts. Maybe is not a thought at all, because in the flow of the memories and in my artworks, I actually try to go also beyond the thoughts, and that's why I use a lot of this white space and spirals, like to suggest a form of space for enlightenment in a way.
The fact that you're using the process of acid washing some of the paper and building your own paper, you open up the same way that synapses can connect in the brain, and you don't really control. You can have thoughts, but you cannot control where your synapses go. You might have the same parallel with the acid wash, where like you build the paper, you're planning on making the paper, but you don't control any fiber, like all of the fiber at once. And so maybe that's also the flowing of the ideas can be seen in that part of the artwork.
Yes, of course. You have to make the projects layer by layer, but then you have to know where your control stops, and the images are open to silence, and the artwork is open to respect this flow.
Thank you for the talk. I'm just curious, from a science perspective, how could quantum measurements on this type of work impact this partnership going forward? How does the next 10 years look?
Whether the quantum world has a particular impact on neuroscience or not, perhaps without delving, too deeply into that here, there are certainly, I think, maybe just as many of these abstract creative ideas between subfields of science that are worth discussing. And it actually might be a really cool avenue to think about how neuroscience might interact with the quantum community here through the lens of art. So I think there's certainly a lot of opportunity there to think about. Absolutely. But where the next 10 years go,
I can talk a little bit more about what quantum technology might enable. So quantum technology right now is at the same stage where the mainframe and the computer were in the 50s. We had this like big machine taking an entire room, capable of barely doing 1 + 1 as a calculation. And then if you had asked these people what this technology will do in the future, I don't think any single of them will have been able to tell you. You'll be able to take photos and then send it to your friends in a tiny box in your pocket. Like I think that's down the line, it would have been impossible. And I think we are basically at the same stage right now where we are building this machine that takes an entire room. And so we think that they will be able to outperform all the classical computers. And so we should be able to do simulation that are not possible in the lifetime of a human. So maybe if you can run your laptop for 1,000 years, maybe you'll end up with the same calculation, but quantum technology, and especially quantum computation, might be able to bring an answer. We want to bring this technology to the public and make sure that everybody can touch that technology and use that, because the same way that when we gave computer to people, they developed the application, that's something we want to do. So if you bring this, and if you give a quantum computer or any other quantum device that can do quantum material, quantum sensing, maybe that's something that the neuroscience community will come in and say, oh, this is exactly what we needed, and that will foster collaboration. But I think it's a little bit difficult to predict right now, but we know that down the line, in the next 5, 10, 15 years, we might be able to use that technology the same way that computers have invaded every lab space in this. I don't know if it's going to be helpful with art, but I think having this lens of seeing how we can use this technology to make something that is counterintuitive and maybe use that technology for something else is something that I think is very interesting.
So where are we going in 10 years?
I agree with you that this field is running in a direction which is unpredictable in a way, and on the other side, through the art, is giving more and more inner visions not just in ourselves and our brains as we are talking tonight, but also within the matter of the reality. And this is very exciting, and I think that both working on this probabilities concept, or the concept of the entanglement, which has been always there in the visual arts, I mean, and also the relationship between the viewer and the object, is something which has been always there. So maybe we are going to see a sort of integration between these fields. I believe so.
Yeah. Well, stay tuned for the next 10 to 15 years. Well, I would like to thank all of you for coming tonight for this event. I would like to also thank my two guests, Mike and Serena, and the Frankie program and the WUSA Institute for hosting us tonight. Thank you all, and have a good night.
Thank you all.
Thank you.